Imagine for a second that you are staring at this massive, sprawling manuscript.
Right, like a really, really thick book.
Exactly. I mean, it is 3 billion letters long. And it's filled with this ancient, incredibly complex code that dictates basically everything about how a machine is built and how it operates.
Okay, I'm with you.
But somewhere deep in that massive text, there's a typo. Just, one single wrong letter out of billions.
Which doesn't sound like a big deal, but
But it is. That one typo causes a devastating, catastrophic failure in the machine. And historically, in the realm of human biology, if you were born with that kind of typo in your DNA, well, that was it.
yeah, it was a genetic fate you really couldn't escape.
Right. But consider what it would actually mean to have a biological word processor, like a tool that allows us to simply search for that specific error in the DNA of a living organism, highlight it, and just press delete.
It's wild to even think about. I mean, it completely shatters the binary idea of biological destiny. We transition from being passive readers of our own genetic material to active editors. We are suddenly capable of rewriting the code of life as it's running.
Which brings us to welcome you to today's deep dive. Our mission today is to explore the life, the scientific breakthroughs, and honestly, the profound ethical dilemmas of Jennifer Doudna.
The American biochemist who co-invented CRISPR genome editing.
Yes, exactly. And we are pulling all of our insights today from a really comprehensive collection of notes and excerpts from her Wikipedia page, which has been updated through early 2026.
It's a great set of source material.
It really is. And, this CRISPR technology operates essentially as the ultimate central plus F and delete for genetic diseases. But to grasp why this is considered like one of the most significant discoveries in the entire history of biology, we really have to look past the tool itself.
We do,
Yeah.
because this exploration is fundamentally about the person who helped unlock this almost unthinkable power to control evolution.
Unthinkable power is a good way to put it.
Yeah, and her trajectory perfectly illustrates the unpredictable, often really messy path of foundational science. A discovery that fundamentally alters humanity's relationship with nature didn't begin with some grand, well-funded quest to cure a specific human disease.
Right. It wasn't a corporate mandate.
Not at all. It began with just a raw, baseline curiosity about how the invisible world is structured.
I really want to trace that curiosity back to its roots, because before you can rewrite the code of life, you have to understand the invisible structures that actually make it up.
You have to know what you're looking at.
Exactly. So let's look at her beginnings. She was born in Washington, D.C. in 1964, but her family moved to Hilo, Hawaii when she was 7.
Quite a change of scenery.
Oh, totally. And growing up in a place like Hawaii, it offers this profound immersion in complex biological systems. She was just surrounded by this incredibly diverse, highly specialized flora and fauna, and that really seems to have tuned her mind to observe how things fit together in nature.
That kind of environment definitely nurtured a very specific kind of systemic observation for her.
Yeah.
But the true intellectual catalyst actually arrived in the 6th grade.
Oh, right. The book.
Yeah, her father, who was an American literature professor, gave her a copy of James Watson's book, The Double Helix.
Which is all about the race to discover the structure of DNA.
Right, and reading that account of untangling these deep molecular mysteries, it just ignited this lifelong obsession with the architecture of the microscopic world.
I just love that a literature professor handing his daughter a book essentially writes her future. It's a remarkable turning point. And she clearly thrived academically after that. She frequently cited her 10th grade chemistry teacher, Jeanette Wong, as a major influence too.
A good teacher changes everything.
They really do.
And-
Yet when she goes off to Pomona College in California to study biochemistry, she actually hits a wall.
Yeah, this part always surprises people.
It's crazy. During her freshman year, sitting in general chemistry, she has this complete crisis of confidence. She actually considers abandoning science entirely so she can switch her major to French.
To French.
Yeah.
I mean, the fragility of early academic trajectories is always so striking to me.
It really is.
Here's someone who will eventually alter the course of human genetics. And she was on the verge of just walking away because she doubted her own aptitude.
And the irony is, it was her French teacher who recognized her scientific drive.
Yeah, the French teacher was the one who advised her to stick with biochemistry.
It just makes you wonder how many scientific revolutions have been lost to freshman year imposter syndrome,
Probably a lot, sadly.
Fortunately for us, she persists. She earns her PhD at Harvard under Jacques Ostek. And then she makes this really fascinating pivot. Instead of focusing on DNA, the famous double helix that sparked her interest in the first place, she zeroes in on RNA.
Which is a huge distinction. DNA gets all the glory as the master blueprint locked away in the nucleus. But RNA is the molecule that is actually out there on the construction site doing the work.
Yeah, she was specifically looking at these things called ribozymes
Hmm.
because I think most people, if they know about RNA, they think of it as just a passive messenger. Like it just carries the DNA code to the cellular machinery.
Right, messenger RNA, sure.
But ribozymes are RNA molecules that fold up and actually act like enzymes. They are physical machines that cut and splice other molecules.
And understanding those machines became the defining challenge of her early career. Because there was a foundational rule in biology. You have to understand form dictates function.
Form dictates function. Okay, break that down for us.
Well, you cannot understand how a molecular machine works, much less how to manipulate it or build a new one, until you can see its exact physical shape.
You have to see the gears to know what the machine does.
Exactly. Proteins, for instance, fold into these very complex 3D structures around a core of water-repelling amino acids. Doudna suspected that RNA could also form these intricate, stable, three-dimensional shapes to perform its enzymatic work.
But she had to prove it.
Right. Proving it required visualizing the molecule at an atomic level.
Which brings us to a massive bottleneck in her research. Because she moves across the country to work with Thomas Seck in Colorado and later takes a position at Yale, and she spends years, literally years, trying to capture a three-dimensional image of an RNA molecule using X-ray crystallography.
Which is incredibly tedious work.
Now, let's just pause on the sheer difficulty of this. You have to force these microscopic, naturally floppy RNA molecules to form a perfectly ordered, rigid crystal, and then you shoot X-rays through them to map where the atoms are.
Right.
It's kind of like trying to perfectly stack millions of identical cooked spaghetti noodles.
That is a perfect analogy. Cooked spaghetti does not want to form a rigid structure.
No, it doesn't. And she started this project in 1991 and didn't get the final structure until 1996. 5 years just to take a picture.
But when she finally solved the structure, it was a total revelation.
What did she actually see when she got the picture?
Well, she discovered that this specific catalytic RNA folded around a core of exactly 5 magnesium ions. It was this elegant, incredibly complex architecture.
Oh, wow. So the magnesium ions are like the central tension pole of a complex circus tent, right? Like holding the entire elaborate structure in place.
That's exactly it. Prior to this, the scientific consensus heavily favored proteins as the primary complex machines of the cell. Doudna's structural map was a massive breakthrough because it provided undeniable physical proof that RNA was capable of sophisticated structural folding, totally independent of proteins.
So if you think about trying to pick a highly complex lock in the dark, you can jam tools in there all day and maybe get lucky, but you don't really understand what you're doing.
Right, you're just guessing.
Doudna essentially turned the lights on. By mapping those 5 magnesium ions and the folded structure, she allowed scientists to actually see the pins inside the lock.
Beautifully put.
So she figures out the shape of RNA, she's promoted to a full professor at Yale, does a stint at Harvard, and then in 2002 moves to UC Berkeley. Her husband, Jamie Kate, was also there researching biofuel yeast.
Yeah, they were both well established.
Very established. She is firmly known as a master of basic structural biology. So how does a structural RNA expert find herself at the center of a revolutionary gene editing tool?
Well, the pivot hinges on the modern internet, surprisingly enough, and the open, collaborative nature of a university campus.
Okay, I love this part of the story.
Yeah, in 2006, another Berkeley scientist named Gillian Banfield was studying microbial communities. She was deeply interested in a phenomenon called RNA interference.
Which is what, exactly?
It's essentially how RNA molecules can intercept genetic messages and silence specific genes. Banfield wanted to know more, so she literally typed RNA interference in UC Berkeley into a Google search to find a local expert.
And Jennifer Doudna's name populated right at the top of the results. The idea that the genesis of the most powerful genetic engineering tool in human history traces back to a random Google search is just astounding to me.
It really is. So Banfield reaches out, they meet, and Banfield introduces Doudna to this bizarre repeating sequence found in the DNA of bacteria called CRISPR.
Now, this is right around the time Doudna is actually tempted to leave academia entirely, right?
Yeah, in 2009, she actually takes a leave of absence from Berkeley to lead discovery research at Genentech, which is a massive biotech corporation.
But she quits after just 2 months.
Just 2 months. It seems she realized the corporate environment, which is naturally highly driven by immediate commercial applications, wouldn't offer her the freedom to obsess over a seemingly random biological oddity like CRISPR.
So she cancels her obligations, leaves the corporate money, and rushes back to her lab at Berkeley.
She was entirely consumed by the puzzle of it all. Together with French microbiologist Emmanuelle Charpentier, Doudna began dissecting exactly what these CRISPR sequences were doing.
And what they uncovered in their landmark 2012 paper was the physical mechanism of a billion-year-old evolutionary arms race between bacteria and viruses.
Which is wild because,
Yeah, I always thought of bacteria as the microscopic threats we needed immunity from. The concept that bacteria themselves have an immune system to fight off their own infections, these viruses called bacteriophages, is fascinating.
it is, and the mechanism is brilliant.
From what I gather from the notes, it works kind of like a molecular mugshot system.
A mugshot system, yeah, exactly.
Like when a virus attacks a bacterium, if the bacterium survives, it snips out a tiny piece of the virus's DNA and physically inserts it into its own genetic code, specifically in this CRISPR region.
Right, it literally updates its own genome to keep a permanent record of its enemies.
Which is just a remarkably efficient way to survive.
It is. And the next time that specific virus invades, the bacterium accesses that genetic archive. It creates a small piece of RNA, a so-called guide RNA, that perfectly matches the viral mugshot.
Okay, the guide RNA.
Yeah. This guide RNA then bonds with a protein called Cas9. Cas9 is essentially a pair of molecular scissors.
I see. So the guide RNA acts as the GPS.
Exactly. It hunts through the cell until it finds the invading viral DNA that matches the mugshot. Once it finds a perfect match, Cas9 unwinds the viral DNA and cleanly severs both strands, instantly neutralizing the threat.
Okay, that perfectly explains how bacteria defend themselves in nature. But observing a natural phenomenon is very different from creating a biotechnology.
That is a huge leap.
Right. So what did Doudna and Charpentier actually do to turn this bacterial defense mechanism into the biological word processor we talked about at the start?
Well, they isolated the components and proved that the system was entirely programmable. They demonstrated that you didn't have to rely on the bacterium's natural guide RNA.
You could make your own.
Exactly. You could synthesize a custom guide RNA in a lab, attach it to the Cas9 scissors, and drop it into a test tube. The Cas9 would blindly follow your custom coordinates and cut whatever specific sequence of DNA you programmed it to find.
So Ctrl plus F to find the exact sequence you typed in, and Cas9 acts as the delete key. And because DNA is the universal language of life on Earth, this tool should theoretically work on any organism, right?
Which is the ultimate vindication for curiosity-driven research.
How so?
Because nobody secures a billion-dollar grant to cure human genetic diseases by studying how obscure bacteria fend off viruses in a random puddle of water.
That's a great point.
They simply ask, how does this complex shape work? And in doing so, they unlocked a mechanism that reshapes our entire approach to genetics.
But the purity of that scientific discovery didn't last very long, did it?
No, it definitely did not.
The moment that 2012 paper was published, proving the system was programmable, the reality of modern biomedicine took over. We go straight from the sterile lab bench to the courtroom and the boardroom.
Oh yeah, the race to own the code of life kicked off a truly brutal patent war.
Right, so Doudna and her UC Berkeley collaborators immediately filed for a patent on the CRISPR-Cas9 technique. However, just months later, another team, led by Feng Zhang at the Broad Institute, which is affiliated with MIT and Harvard, published a paper showing they had successfully used CRISPR-Cas9 to edit genes inside cultured human cells.
And they filed their own patents.
So the distinction here between editing DNA in a test tube and editing it inside a human cell became the focal point of this massive ongoing legal battle.
It really did. And let's unpack the biology of that legal argument, because it's not just a technicality.
Yeah, please do.
See, bacterial DNA is essentially floating around loose inside the cell. But human cells, eukaryotic cells, are vastly more complicated. Our DNA is locked tightly inside a nucleus, and it's wrapped around complex proteins, like a densely coiled spool of thread.
That sounds like a nightmare to navigate.
It is. Navigating a cutting tool through the cellular membrane into the nucleus, unpacking the thread, making a precise cut without destroying the cell, and tricking the cell into repairing the cut the way you want it to. That is a monumental biological hurdle.
So the Broad Institute's legal argument rested precisely on that complexity. They argued that their work wasn't merely an obvious extension of Doudna's discovery, but a massive inventive leap necessary to make the technology clinically relevant for humans.
Right. And UC Berkeley countered that their foundational patent application covered the fundamental mechanism. They said they had explicitly outlined how it could be applied to eukaryotic cells, making Zhang's work just an expected progression.
It feels akin to the invention of the internal combustion engine. Like
Yeah.
Doudna and Charpentier conceptualized and built the engine on a workbench, proving it could spin and generate power.
That's a great way to look at it.
And then the Broad Institute figured out how to drop that engine into a highly complex sports car and successfully drive it down the highway.
Both are incredible feats.
Right. But the courts had to decide who owned the rights to the car itself. And in the U.S., the courts largely sided with the Broad Institute, ruling that proving the application in human cells was distinct enough to warrant a separate patent. Meanwhile, UC Berkeley won victories in European courts due to procedural issues regarding who the Broad Institute listed on their applications.
So the landscape fractured globally. And, it raises an enduring question of the engine of modern science.
Which is?
Well, developing a CRISPR-based genetic therapy and running it through the FDA's rigorous clinical trial process costs hundreds of millions, sometimes billions of dollars. Private companies will not invest that capital without the exclusive commercial protections that patents provide.
That makes sense.
But conversely, a fiercely litigated multi-year patent war risks locking up a transformative technology.
Yeah.
It could potentially terrify other researchers away from the field entirely due to the threat of catastrophic lawsuits.
Yeah. Does legal combat speed up innovation or stifle it? It's tough. But Doudna's reaction to all this commercial fallout is super revealing.
She didn't just sit on the sidelines.
No, she co-founded several companies to push the technology forward. Caribou Biosciences, Intellia, Scribe, Mammoth Biosciences. She even briefly co-founded a company called Editas Medicine alongside Feng Zhang, her chief rival.
Though she left that venture in 2014 after what was described as a highly tense, quote-unquote, divorce-like environment.
Yeah, I can only imagine the tension in those meetings. What stands out to me, though, is that she didn't allow the patent litigation to paralyze her actual scientific output. Just look at her actions during the 2020 COVID-19 pandemic.
Oh, that was a huge moment for her lab.
She pivoted her team at the Innovative Genomics Institute, setting up a massive testing center that processed over 500,000 patient samples from the local community. Furthermore, her company, Mammoth Biosciences, utilized CRISPR technology to validate a rapid diagnostic test that identified the virus faster and cheaper than standard PCR tests.
She actively weaponized her laboratory resources to address a real-time global crisis. She didn't retreat into the legal battles or the ivory tower. She focused on immediate, tangible application.
Which is incredible leadership.
It is. And it brings us to the ultimate and perhaps most difficult chapter of her ongoing legacy. It's one thing to uncover the tool and another to navigate the intellectual property.
Right.
But the heaviest burden Doudna carries is her role in navigating how humanity should wield this profound power.
Because the medical promise we are looking at is staggering. We are talking about utilizing CRISPR to treat diseases that have haunted human biology forever. Sickle cell anemia, cystic fibrosis, Huntington's disease, even targeting the DNA of HIV hidden in human cells.
The potential to alleviate generation-spanning suffering is immense.
It really is. But we've essentially handed humanity a loaded evolutionary weapon.
And she recognized that gravity immediately. In 2017, Doudna co-authored a book called A Crack in Creation. It's highly unusual for the architect of a major scientific paradigm shift to write a book aimed at the general public that essentially serves as a massive caution flag.
Yeah, inventors usually just hype their stuff up.
Exactly. Yeah. But in it, she draws a very hard, non-negotiable ethical line regarding how this technology should be applied.
She draws the line between somatic gene editing and germline gene editing.
That is the crucial distinction.
Let's break that down for our listeners. With somatic editing, you are treating the living patient. Take sickle cell anemia, for example. Doctors can extract blood stem cells from a patient, use CRISPR in a lab to find the typo causing the sickle shape, delete it, replace it with the correct sequence, and infuse those corrected cells back into the patient.
And the patient is cured?
The patient is cured. But their reproductive cells, their sperm or eggs, remain completely untouched. If they have children, they can still pass the original sickle cell gene down.
Germline editing, on the other hand, involves using CRISPR to alter the DNA of a human embryo.
Which changes everything.
It does, because if you edit an embryo, you are not just treating a single patient. Every single cell in that resulting human being will carry the edit, including their reproductive cells.
Oh, wow.
That means the genetic change you introduce will be permanently woven into the human gene pool. It gets passed down to their children and their children's children into perpetuity.
It is the biological equivalent of pushing a permanent software update to the entire human race, and there is no rollback feature.
No beta test, no taking it back.
Once those edited genes enter the breeding population, they are there forever. The potential for unintended consequences down the generational line is terrifying. And beyond safety, there is the immediate specter of enhancement.
Right. Designer babies.
Using the technology not to cure a lethal disease, but to engineer traits. Doudna was so alarmed by this prospect that she led a high-profile call for a global moratorium on any clinical application of germline editing.
Which was a bold move.
It was, but I have to push back on the reality of that approach. Is a voluntary international moratorium really going to stop a rogue laboratory or some unregulated clinic from offering designer babies to the highest bidder? The technology is relatively cheap and widely understood now.
That vulnerability is real. And the history of science suggests that voluntary pauses are incredibly difficult to enforce globally. A moratorium is, at the end of the day, a piece of paper, not a physical barrier.
Exactly.
But we must evaluate the significance of Doudna's posture. Historically, the pioneers of world-altering technologies, whether the splitting of the atom or the architecture of the internet, often release their creations into the wild and claim a kind of passive neutrality regarding how society uses them.
They wash their hands of it.
Right. But Doudna rejected that abdication of responsibility. She stepped directly into the ethical crosshairs, demanding international oversight and governance of her own life's work.
She forces the scientific community to acknowledge a very uncomfortable truth.
Yeah. She forces them to see that the distinction between somatic and germline editing isn't merely a biological boundary. It is the absolute threshold between practicing medicine and actively directing the future evolution of our species.
Practicing medicine versus directing evolution. The weight of that is just hard to process. And the scientific community has certainly recognized the magnitude of her contributions.
Oh, without a doubt.
In 2020, she shared the Nobel Prize in Chemistry with Emmanuelle Charpentier. By 2023, she was inducted into the National Inventors Hall of Fame. In 2026, she was elected to the National Academy of Engineering.
All massive honor.
But perhaps the most poetic honor, in 2025, the Lawrence Berkeley National Laboratory unveiled a massive new supercomputer specifically designed to model complex biological systems, and they named it Doudna.
An incredibly fitting tribute for a scientist whose entire career was dedicated to mapping and decoding the complex data structures of the natural world.
Let's pull all of this together.
Yeah.
We followed a journey today that started with a young girl reading a book about DNA in Hawaii, moved through a crisis of academic confidence in college, and culminated in a Nobel laureate who holds the keys to human evolution.
It's an amazing arc.
But the core insight from this massive stack of sources is really the true nature of scientific progress.
Right.
The ability to potentially cure sickle cell disease, or Huntington's, didn't originate in a pharmaceutical boardroom with a strategic plan to cure genetic ailments. It originated from a pure, unadulterated desire to figure out how a tiny, seemingly insignificant piece of RNA folded itself into a specific shape inside a test tube.
It came to the basic human drive to ask, how does this work?
Yes, the downstream applications of foundational science are entirely unpredictable.
Absolutely. By simply trying to see the invisible structures of the microscopic world, she uncovered a mechanism that fundamentally redefines the limits of biology.
Which leaves us with a fascinating, lingering thought for you to ponder. If the most powerful genetic engineering tool in human history was just sitting there, quietly operating inside a common bacterial defense system for a billion years.
Just waiting.
Yeah, just waiting for an internet search to connect the right curious minds. What other unimaginable, world-altering superpowers are currently hiding in the dirt of our backyards, just waiting for someone to look close enough to actually see them?